Hello everyone, and welcome to a new episode and a new series. I'm starting a series today that will explore the diversity of life on planet Earth, and I'm beginning with the smallest, oldest life that exists, the Archaea and the bacteria. These microscopic organisms cover every living thing, every plant, every animal, both inside and outside. You have bacteria and Archaea on your skin, in your mouth, and in your intestines. They exist in soils, in swamps, and in the ocean, even in extreme environments like hydrothermal vents and super salty lakes. Archaea and bacteria cover the planet, and they're so fundamental that no complex life could readily exist without them. If you listen to the series on the evolutionary history of life on Earth, you should remember that in the first episode I introduced the Precambrian era. This era lasted for nearly 4 billion years, which represents the vast majority of geologic and evolutionary time. This episode will be kind of like that. Bacteria and Archaea represent the vast majority of life on Earth. Of all the world's species, a majority are bacteria and Archaea. Of all the individual organisms on Earth, the overwhelming majority are bacteria and Archaea. Of all life that's ever existed, the oldest, the most fundamental, and the most widespread are bacteria and Archaea. It's because bacteria and Archaea are so important, so universal and ever-present in every ecosystem on the planet, that I really had no choice but to start this series with them. So life is split into 3 domains, and the domains are the largest groups of organization in the tree of life. The first domain that's believed to have existed was the bacteria, and shortly after that emerged the Archaea. The third domain is the eukaryotes. The eukaryotes are organisms with internal organelles, a higher complexity. Many of them are multicellular. Every single animal, plant, and fungus are eukaryotes, as are all of the protists, but I'll cover all of these things in subsequent episodes. Today is all about bacteria and Archaea. So with that said, let me start with the basics, with how these 2 domains are similar. All bacteria and all Archaea are microscopically small unicellular organisms, tens to hundreds of times smaller than a typical animal cell. Bacteria and Archaea are both prokaryotic, which means they don't keep their DNA inside a nucleus. Where eukaryotic cells have a membrane-bound nucleus in the heart of their cytoplasm that encloses their DNA, the prokaryotic cells, like Archaea and bacteria, have nothing. Their DNA molecules form circular loops, which float free and unprotected within their tiny bodies, where they're more accessible to regulatory proteins but more vulnerable to mutating factors like UV radiation. Bacteria and Archaea both reproduce asexually. Because they live lives measured in minutes, they can reproduce dozens of times in a day. This can quickly produce trillions of descendants, each one a clone of its parent, but with species emerging through an accumulation of mutations over thousands of generations in some particular lineage. In this way, bacteria and Archaea are a ridiculously diverse domain of life. There are around 30 recognized bacterial phyla, but more than 50 if candidate phyla are taken into account. Estimates as to the total range of the bacterial domain reach close to 1,000 phyla, although this estimate is based on the fact that new species are discovered all the time. The Archaea are themselves divided into 4 phyla, although there are candidate phyla here as well. 2 of the Archaea phyla are really extensive, with huge extended classes and families with dozens and dozens and dozens of species within them. The other 2 phyla are much less researched, and much less is known about them or the lineages within them. Despite their asexuality, bacteria and Archaea can cooperate in a really unique and fascinating way, called horizontal gene transfer. When horizontal gene transfer happens, 2 cells, be they 2 bacteria or 2 Archaea, or a bacteria and an Archaea, they come in close proximity, and if they can, they extend their cellular membranes in such a way as to meet and produce a tube of membrane that connects the 2 cells. And through this tube, they pass plasmids, or little circular bits of DNA. In this way, even though they're not sexually reproducing, they can exchange DNA, they can exchange alleles. This non-traditional method of genetic information transference has led to the complication of the taxonomy of both of these domains. Because of this, it's really hard to tell when a new allele or a new speciation event or something happened because of a natural mutation or if it happened because of one of these horizontal gene transfer events. It really kind of muddies up the water of the evolutionary history of these two domains. If you were to go out into the field and find any organism, be it some species of grass or tree or an insect or a rabbit, or literally any kind of large complex life, and if you were to take a tissue sample or a blood sample or something and study under the microscope, you would find hundreds, if not thousands, of unique species of bacteria and archaea. They literally cover everything, inside and out. Some of these microscopic species would be known and cataloged, but most would be undiscovered. Most would be species of bacteria or archaea that you would only find in or on those particular plants or animals. For example, your mouth has over 700 known species of bacteria and archaea, and your intestines have over 1,000. After 4 billion years of evolution, the perpetual divergence and speciation of all of these bacteria and archaea has created uncountable numbers of species, all shaped by evolution for an uncountable number of habitats, be it in the soil, on the bark of a tree, on your skin, in your intestines, in your mouth, or anywhere else. Not only will your tissue sample have thousands of bacterial and archaeal species, there will be millions, if not billions, of individual cells. Because these cells are so small, places like your intestines hold many times more bacterial cells than there are your own cells in your own body. If you go outside and grab a handful of dirt, you'll be holding millions of cells of bacteria and archaea. Go to the ocean and with your hands scoop up some seawater. You'll be holding millions of cells of bacteria and archaea. Huge masses of bacteria and archaea exist deep into the floors of the oceans. They exist in hydrothermal vents, in salt lakes and hot springs, across the rugged floor of the world's forests and jungles, and in all the living things of the world. The bacteria and archaea share several major similarities, including their extremely small size, their cell walls, their shared lack of organelles in the nucleus, their ubiquity in virtually every environment, and the important role they play in their local ecologies. But aside from these, there actually aren't too many more similarities. When you zoom in to the molecular level, the differences between bacteria and archaea become much more apparent. For example, they possess different kinds of biomolecules in their cell walls and plasma membranes, and they use different kinds of proteins to replicate and express their DNA. I'll describe a few more of these similarities and differences in greater detail. When I said that bacteria and archaea were small, I don't think small was a strong enough word to describe it. These microorganisms vary in size among themselves, but all of them are absurdly small, as small to your own cells as a wart or a pebble is to you. Bacteria and archaea are measured in micrometers, which are units 1/1,000,000th the size of a meter, or more than 25,000 times smaller than an inch. The smallest bacteria are less than half a micrometer in diameter, and the smallest archaea are 1/10 of a micrometer, while the largest of them all can be up to 100 micrometers wide. The shapes they take vary as much as their size. Some shapes are really common, while others are found in only a small handful of species. Some of the most common shapes are spheres, rods, or spirals, but they can also be shaped like strings, like cubes and tetrahedrons, or like weird kidney beans, flat plates, and other irregular objects. Some archaea that lack a cell wall are shaped like amoeba, like a tomato splattered against a wall. Most species tend to exist as independent single cells, but others form distinctive groups or superstructures. The most common arrangements are simple pairings or groupings of a few cells. The spherical bacteria are called cocci. In pairs, they're called diplococci, and in long chains, they're called streptococci. If you ever had strep throat, you've been infected by a Streptococcus pyogenes bacteria, which under the microscope looks like a bunch of small spherical dots arranged in a squirming line. These little guys infect your throat and your tonsils in their millions, spreading toxins that aggravate and inflame the soft tissues. Coughing or even speaking carries air across the inflamed tissue, and it causes the pain that people with strep throat are all too familiar with. Other cocci bacteria like Staphylococcus or staph form random and irregular clusters that are described as looking like a bunch of grapes. Bacterial species called Sarcina form cubes composed of 8 spheres all mushed together. Species of Archaea can take even stranger forms, linking together into long filaments or giant masses. Despite their often similar shapes, bacterial and archaeal cell walls and plasma membranes have very different chemical compositions. Bacteria possess a chemical in their cell walls called peptidoglycan. Those bacteria with a high concentration of peptidoglycan are called gram-positive, while gram-negative bacteria tend to have just a thin layer of peptidoglycan hidden beneath an external membrane. The classification is a reference to how the bacteria appear under a microscope, after they've been soaked in a purple dye called a gram stain. The gram stain sticks to the peptidoglycan, which makes gram-positive bacteria appear a dark purple. Gram-negative bacteria don't have that much peptidoglycan in their chemical coat, so they don't stain very darkly, if they stain at all. The gram stain test is useful for medicine and healthcare, because if we know what the cell wall and the membrane are like, we can figure out ways to disrupt it or penetrate it to kill the bacteria and cure the infection. The Archaea don't have any peptidoglycan in their cell walls at all. Well, 99% of them don't. There's a group of methanogenic Archaea who have peptidoglycan in their cell walls, but the peptidoglycan molecules they use are modified and quite dissimilar from those found in bacteria. These molecules are called pseudopeptidoglycan, and they possess a different sugar, a different kind of linkage connecting those sugars together, and some of the molecules have a different kind of chirality. Chirality is something I believe I touched on in the first series, although I might have skimmed over it. In either case, the chirality of a molecule describes its orientation. You see, complex molecules like proteins have big, bulbous, irregular, asymmetrical shapes, but they can exist in two orientations, each one a mirror image of the other. They aren't perfectly identical copies, they're mirror images, much like your face in a photograph and your face in a mirror, or like your left and right hands, or your left and right feet. But instead of left and right, the orientations of a chiral molecule are called L and D. In bacteria, their peptidoglycan has D amino acids, but Archaea have L amino acids. The overwhelming majority of amino acids in all living things have the L orientation. Amino acids with the D orientation are much more rare, existing almost exclusively in bacterial cell walls. Chirality isn't the only thing differentiating the biomolecules inside Archaea and bacteria. The molecules they use for respiration, for metabolism, for eating and digesting, huge portions of these molecules are entirely different between the two domains. Internal biochemistry is really where the differences between Archaea and bacteria become obvious. Before I dive into the smallest details, I should start with more general differences and work my way down the chain of specificity. So, most generally, bacteria and Archaea can be organized based on how they produce ATP for energy. Phototrophs make energy through the absorption of light. The chemoorganotrophs make energy through the oxidation of energy-rich organic compounds. And the chemolithotropes make energy through the oxidation of energy-rich inorganic chemicals. Furthermore, bacteria and Archaea consume the carbon they need to build biomolecules through two processes. Autotrophs get their carbon by breaking down simple molecules in the atmosphere, like carbon dioxide or methane, while heterotrophs get their carbon by decomposing the organic molecules produced by other organisms. This is important to understand, because every species alive needs to produce energy and gather carbon, not just bacteria and Archaea. Because there are 3 ways to produce energy and 2 ways to get carbon, this means that there are 6 combinations that life can use. All eukaryotes, from plants to animals, only use 2 of these combinations. Photosynthetic organisms like plants and algae are photoautotrophs, getting their energy from sunlight and their carbon from CO2 in the atmosphere. Animals are chemoorganoheterotrophs, getting their energy and carbon by digesting the tissues of other organisms. In contrast, bacteria and Archaea can utilize all 6 combinations of energy production and carbon acquisition. Because bacteria and Archaea were the first life forms, they evolved the first biochemical pathways, the first iterations of photosynthesis and cellular respiration and fermentation. With the billions of years of evolution that followed, these processes were shaped by mutations and natural selection, creating a wide diversity of species able to adapt to an equally wide diversity of habitats and ecological niches. Alright, back to the 6 combinations. This is going to sound intense, but it's not as bad as it sounds. I'll break it down, but these 6 combinations of energy production and carbon acquisition are the photoautotrophs and photoheterotrophs, the chemoorganoautotrophs and the chemoorganoheterotrophs, and the chemolithoautotrophs and the chemolithoheterotrophs. That was a lot of word salad that I just kind of threw at you, But don't worry, like I said, it's not as complicated as it sounds. So let's start simple, with the photoautotrophs. The prefix photo means they produce energy through absorbing sunlight. All the phototrophs, therefore, live in habitats where they're exposed to the sun. Those that live in lakes and oceans and stuff exist in the photic zone, where sunlight can still penetrate deep enough into the water to support life. Photoautotrophs are those sunlight-dependent prokaryotes that gather their carbon from the CO2 and methane of the atmosphere, or whatever substrate they live on. Cyanobacteria are a famous example of a photoautotroph. They live in lakes and oceans and soils, using sunlight to power their oxygenetic photosynthesis, and they reduce carbon dioxide for the carbon necessary to grow and reproduce themselves. Cyanobacteria exhale molecular oxygen as a result of this CO2 reduction, and the endless trillions of individual cyanobacteria breathing out oxygen over billions of years has led to an oxygenation of the atmosphere. The cyanobacteria produce the vast bulk of oxygen you and I are breathing right now. Take a breath. You were only able to do that because of cyanobacteria. The trees helped a little bit, but the cyanobacteria really did most of the work to give you that oxygen that you just breathed in. The photoheterotrophs, on the other hand, cannot reduce carbon dioxide. Instead, they rely on the consumption of organic materials for their carbon. This means anything from the absorption of carbohydrates and fatty acids in soils and sands to feasting on the decaying tissues of dead or diseased organisms. Next are the chemoorganotrophs and the chemolithotrophs. Both of these groups produce their energy not from sunlight, but through the extraction of electrons from, respectively, organic and inorganic matter. These cells take high-energy electrons from their donor molecules and shuffle them from enzyme to enzyme as energy is extracted through chemiosmosis to produce ATP. At the end of this transport chain, the electron has been reduced to a low-energy state and transplanted onto an acceptor molecule. In eukaryotes, this process takes an electron from an energy-rich molecule like glucose, it produces water as a byproduct, and the electron is finally given to an oxygen atom. Many species of bacteria and archaea, called organotrophs, use glucose and oxygen just like most eukaryotes. But other groups of bacteria and archaea use different molecules and atoms and stuff for this process. For example, the methanogens take electrons from molecular hydrogen and give them to carbon dioxide, producing methane as a byproduct. The sulfur bacteria take electrons from hydrogen sulfide, often pulling an entire hydrogen atom along with the electron, and they put them on the molecular oxygen to create water molecules. The iron reducers take electrons from organic molecules and dump them onto iron atoms, literally reducing the iron with the added electrons. The nitrate reducers do the exact same thing, but instead of iron, they dump their electrons on nitrate, or NO3, to produce nitrogenous byproducts like N2O and molecular nitrogen. There's a tremendous diversity in the chemical foods that bacteria and archaea use for energy. Because of the huge diversity of chemicals that bacteria and archaea can live on and feed off of, they can exist everywhere. They can consume the methane in your gut, or from a peat field. They can consume sulfur in a deep-sea vent, or from volcanic soils. They can persist almost anywhere, altering virtually any habitat with their consumption and respiration. Earlier, I said that cyanobacteria were responsible for producing most of the oxygen in the Earth's atmosphere, but I left out the fact that all kinds of bacteria and archaea are responsible for changing the chemical compositions of the oceans, enriching the world's soils, supporting the biochemistry of more complex organisms, and influencing the fundamental evolution of life itself. When we talk about bacteria and archaea, we're talking about literally the most fundamental species, the most deeply rooted in the world's ecologies. One of the most drastic effects that bacteria and archaea have had on the world was the oxygenation of the atmosphere. I discussed it briefly in episode 25 on the Precambrian eon, but basically this event was an accelerating catastrophe. As the cyanobacteria produced molecular oxygen, it filled the atmosphere. At first, this reactive molecular oxygen bound to iron and organic materials, but it was only a matter of time until these oxygen sinks were full. When the world's exposed iron and decaying biological material couldn't bind any more oxygen, the excess began accumulating in the atmosphere, with nowhere else to go. The cyanobacteria kept breathing out oxygen, and it kept accumulating until it got to dangerously high concentrations. Oxygen was toxic to most life alive at the time, and the rise of oxygen in the atmosphere killed off most life in what's called the oxygen holocaust. Despite the fact that this oxygen killed off a lot of the life present at the time, it allowed the surviving life forms to adapt to a world saturated with oxygen. It opened the door for life to incorporate oxygen into their biochemistries, to use the electronegative oxygen in the electron transport chains. Because oxygen has a really high free energy value as an electron acceptor, it allowed newly adapted life to produce significantly more chemical energy than before. This abundance of energy would have supported the existence of larger, more complex life forms, like the first giant arthropods and the dinosaurs and us. Bacteria and archaea also help sustain the world's forests and jungles and agriculture through a phenomenon called nitrogen fixation. You see, plants use nitrogen as an integral part of their biochemistries. Everything from the chlorophyll to their structural tissues to their biomolecules uses a lot of nitrogen. As a result, the growth of plants, and thus the growth of forests and jungles and crops, is often limited by a lack of nitrogen. The plants will grow as much as they can, with as much nitrogen as they can. But when nitrogen is rare or difficult to access, the growth will slow to a stop. For example, most of the atmosphere is molecular nitrogen, N2, but this nitrogen is largely inaccessible. The two nitrogen atoms are bound together with a triple bond, which almost no plant or algae can break apart. Bacteria and archaea are so important because they actually do have the ability to break apart this bond. The lineages of bacteria and archaea that can fix nitrogen have an enzyme called nitrogenase, which is the critical enzyme in breaking N2 into nitrate and ammonia. Unfortunately, nitrogenase can't be exposed to oxygen. If the enzyme comes into contact with oxygen, it gets poisoned and ruined. Because of this, those life forms with nitrogenase are confined to anaerobic environments, to habitats devoid of any oxygen. As a result, these nitrogen-fixing bacteria and archaea live in shallow water, where they feed bioaccessible nitrogen into the roots of various plants. These kind of microbes are often used in rice paddies to pump nitrogen into the water, enriching the plants and bolstering their growth. Nitrogenase-carrying bacteria and archaea also live on dry land, despite being surrounded by the oxygen in the atmosphere. They pull this off by forming symbiotic relationships with plants. The nitrogen fixers are literally encapsulated in nodules along the roots of the plants, and within these small nodules, the nitrogenase is kept in an oxygen-free environment, where it can fix nitrogen for the plant in safety. The nitrogen-fixing activity of these bacteria and archaea has enabled a nitrogen cycle, which is kind of like the water cycle, but it involves animals instead of rain clouds. The nitrogen cycle begins with the molecular nitrogen in the air. The molecular nitrogen gets fixed by bacteria and archaea into nitrogen-containing biomolecules. These biomolecules can be decomposed by more bacteria and archaea, and by some species of fungus into ammonia and ammonium ions. Plants incorporate nitrogen from these sources into their bodies, and the animals that eat the plants incorporate that nitrogen into their bodies, as do the predators who eat them. When all these plants and herbivores and predators die, their bodies are decomposed, and the nitrogen is fixed into ammonia. Bacteria and archaea can oxidize the ammonia into nitrite, which can be further oxidized by some bacteria into nitrate. This nitrate can be used in plants, where it goes back into the cycle, or it can be reduced back into molecular nitrogen and put back into the atmosphere, where it then starts the cycle all over again. This prokaryote-enabled nitrogen cycle is crucial. Without it, we wouldn't have the forest and jungles that we have. Plants couldn't be fertilized, and crops would be stunted and give smaller yields. The nitrogen cycle is clearly the most widespread and important chemical cycle for plant life, but bacteria and archaea are also involved in cycling chemicals like sulfur, phosphorus, and carbon. It's not all sunshine and roses, however, as human interference in the nitrogen cycle through artificial fertilizers and industrial agriculture often leads to nitrogen pollution. This is a complex problem that affects life at many levels in an ecosystem, so try and follow along with the path of pollution that nitrogen takes. An average plot of farmland gets saturated with nitrogen in the fertilizer, which fuels the growth of the bacterial and archaeal communities in the soil. A lot of the nitrogen in the fertilizer gets converted by these microbes into nitrate, and the nitrate, being soluble in water, is easily flushed out of the soil through rainfall, or just by the farmer watering the farmland. Unfortunately, these nitrates accumulate in rivers and groundwater sources like wells and aquifers, and they get funneled into the oceans. The nitrates are consumed by bacteria and archaea in the oceans as food, and their populations explode. More food means you can support more people, and this applies to bacteria and archaea just like it applies to us and everything else. When the swollen populations reach maturity and die, their microscopic corpses litter the ocean floor in their trillions. On the ocean floor, even more bacteria and archaea decompose the bodies of their dead cousins, and they experience a similar but delayed boom in their populations. As these seabed-dwelling bacteria and archaea decompose the organic matter and reproduce, they consume an absurd amount of oxygen. And this is where stuff gets bad, because the massive overconsumption of oxygen in these situations creates massive dead zones in the water, areas deprived of oxygen where fish and other oxygen-dependent organisms can't live. They're literally vast expanses of anoxic water, filled with no life, save for any anaerobic microbes that happen to be floating around. These dead zones can be catastrophic for marine ecosystems and the fishing industry in general. It's like habitat loss in the ocean. The farmland in Central America has nitrates that get flushed into the Mississippi River, and as they flow into the Gulf of Mexico, they've created a dead zone nearly the size of New Jersey. The bacteria and archaea in our intestines can also be thrown out of equilibrium by human interference. The gut flora play a huge role in maintaining the health and vitality of a human, or any other animal, like a horse or a cow or, whatever. The bacteria and archaea in our stomach and intestines helps break down food. They help digest food and make sure as many kinds of biomolecules get broken down as possible. They can even help in areas not directly related to digestion, like supporting the immune system and producing hormones for fat storage. When you consume antibiotics or probiotics, you alter your gut flora. Antibiotics damage and kill bacteria and archaea, and if you take too much, you can destabilize the ecology of your gut flora. This can irritate your bowels, and by killing off the gut flora that boost your immune system, you can actually make yourself more susceptible to diseases. Because bacteria and archaea are involved in the digestion of carbohydrates, their depletion leads to incompletely digested carbohydrates. These carbs soak up water, which can lead to diarrhea. Because of these and other related issues, antibiotics have to be consumed either in careful doses or in combination with probiotics to counteract the negative effects. I'm almost at the end of the episode, and I haven't even mentioned all the diseases directly caused by bacteria and archaea infections. As virtually everyone in the modern world knows, bacteria can infect you and cause disease. They do this by penetrating your body's defenses, like your skin and your immune system, interfering with your cells to compromise your health. This can happen through a huge variety of mechanisms, so many that it would be literally impossible for me to list them all. To barely scratch the surface of this subject, bacteria can infect other things and harm them by releasing toxins that interfere with the proteins of healthy cells, or that disrupt the membranes of organelles, or that interfere with some kind of regulatory process or chemical pathway. These toxins and interfering effects can wound cells and make them unable to function properly, or they can entirely kill infected cells. The immune response to a bacterial infection causes swelling and inflammation, and this can cause more damage to the innocent cells getting involved in the pathogen attack. The immune system fights back by identifying specific chemical tags on the exterior surface of the bacterial cell, and by mounting armed responses to the invaders. That's almost an offensively simplified explanation of the immune system, so I feel obligated to tell you guys that I will cover the immune system in much more detail in a future episode. But with that said, I think that's about all that I have for you today. To recap, bacteria and archaea are the oldest and most fundamental forms of life on Earth. They cover everything, and they live everywhere. They're involved in everything, and just by the nature of them existing and eating and breathing, they literally power the numerous chemical cycles that define life on Earth, from nitrogen cycling to phosphorus to carbon. Although they're all small and they often look similar to the untrained eye, bacteria and archaea are vastly different creatures on a chemical level, with different molecules in their membranes and their cell walls, with different means of producing energy and accumulating carbon to grow and reproduce. Bacteria and archaea are integral for your life and mine. They let us digest our food. They let us stay healthy and happy. So the next time you eat a meal, appreciate the fact that the energy from this food, the energy that keeps you alive, is largely being processed by the hard work of the bacteria and archaea that line your intestines. I hope this episode was fun and informative, and I hope you learned something cool. As always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button or share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.